Biochemistry 401 Lecture 17. We're going to talk about other fuels in glycolysis. These will include galactose, fructose, and glycerol. Now we saw that glycolysis is the breaking down of glucose in 3 stages with 10 reactions. But other fuel sources can also enter into the glycolytic pathway, and we're going to look at 3 prominent fuels. These are galactose and fructose, which are monosaccharides, and glycerol, which is a degradation product from fats. Now, galactose enters the glycolytic pathway as glucose 6-phosphate. And in these slides, I want to draw your attention to the fact that we're going to see glucose 6-phosphate written 2 different ways, as glu 6-phosphate and as G6-phosphate. Galactose comes from lactose. Lactose is a disaccharide, and it is found in milk. This disaccharide is hydrolyzed on the surface of enterocytes to yield galactose and glucose. The second way is through the action of beta-galactosidase that is found in gut bacteria. Now, many persons, especially adults, have insufficient levels of lactase in their enterocytes, and so a lot of the lactose from the diet is broken down by gut bacteria. Now, when gut bacteria begin to break down lactose and use it as a fuel source, this results in the production of gas. And so many people who are lactose intolerant will have a great deal of gut pain and gas. And this is because of an increase in activity in beta-galactosidase in the gut bacteria and an ability to use this fuel source. In persons who have enough lactase, the enterocytes are able to break down this lactose into glucose and galactose and then uptake those monosaccharides and use them as a fuel source. However, if people have insufficient levels of lactase, then this fuel source is given to the bacteria to break down. Now, these 2 monosaccharides, D-glucose and D-galactose, are epimers, and again, epimers have the same chemical formula. However, their configurations are different. They differ at 1 asymmetric carbon only. And we can see in the case of D-glucose and D-galactose that the difference lies in carbon number 4. And so D-glucose and D-galactose are C4 epimers. We can see this more clearly in this diagram. We can see that the hydroxyl at position 4 in glucose is below the ring, whereas this hydroxyl in galactose is above the ring. And so when you see monosaccharides drawn in Fischer projection, it's important to realize that when constituents are on the left, they lie above the ring, and when constituents are on the right, they are below the ring in the Hayworth structure. And so let's walk around the ring for just a minute, using glucose as a model. We're going to start at carbon 1. Now, the hydroxyls at carbon 1 and 2 and 4 are all pointing downward, below the ring, whereas the hydroxyl at position 3 is the only hydroxyl that is pointing up, above the ring. In the case of galactose, we have 2 hydroxyls that are above the ring, those at position 3 and 4. And so again, galactose is a C4 epimer of glucose. So now that we know the difference between glucose and galactose, let's see how galactose is broken down. As with any monosaccharide, if we want to keep it in the cell, the first thing we're going to do is phosphorylate it to trap it in the cell and to help to destabilize it. Here we see galactose on the left, and it is going to be phosphorylated by the enzyme galactokinase. Now, these enzymes that we're going to see are going to be very easy to remember. We have an enzyme that acts as a kinase to phosphorylate galactose, and so it is galactokinase. Kinases are usually given the name for the substrate on which it works. And so galactokinase works on galactose to form galactose 1 phosphate. Therefore, this enzyme is galactokinase. Now we're going to see this pathway written in two different ways to help you to understand what's going on. Now that we've trapped galactose 1 phosphate, we're going to do a little bait and switch. We're going to take galactose 1 phosphate and trade it off for a glucose 1 phosphate. And the donor molecule is going to be UDP glucose. And so all we're going to do is transfer galactose 1 phosphate for glucose 1 phosphate, and that's all we're going to do. And we're going to end up with UDP galactose instead of UDP glucose. And the other product of this reaction is glucose 1 phosphate. And we're going to use this further. But the thing is we still have UDP galactose, and we need to change this so that we can continue on this cycle. And so we're going to epimerize UDP galactose to make UDP glucose, just by switching the position of the hydroxyl at position number 4. And then this UDP glucose can go all the way up to the first reaction and start the cycle all over again. So let's go through this again, slowly. Galactose 1 phosphate plus UDP glucose will react in a reaction that's catalyzed by galactose 1 phosphate uridyl transferase. Now again, this is an enzyme whose name says what it does. We're then going to take the products UDP galactose and glucose 1 phosphate, and we're going to use those further. Glucose 1 phosphate is going to be worked on so that it can enter the glycolytic pathway. But UDP galactose has to be changed so that it can re-enter the cycle. And so UDP galactose is going to be epimerized by UDP galactose 4 epimerase. Again, the enzyme's name says what it does. The product will be UDP glucose, and this can go right back up to step 1. Glucose 1 phosphate will be converted to glucose 6 phosphate, because that's what we need to enter glycolysis. And the enzyme that does this is phosphoglucomutase. Mutases switch groups within the same molecule. So phosphoglucomutase switches the phosphate group from position 1 to position 6 to enter glycolysis. A failure in this pathway results in the condition called galactosemia. Galactosemia results from a high concentration of galactose metabolites in the system, and this includes a high concentration of galactose 1 phosphate. And this is especially dangerous for infants whose diet consists of milk. And so, to make sure that this is caught early, infants are screened for galactosemia. The symptoms of galactosemia include a failure to thrive, vomiting, and liver problems like jaundice, an increased liver size, cirrhosis, irritability, cataract formation, brain damage and kidney damage, and eventually death, if this is not caught. The causes of this condition are recessive mutation that results in a decrease in these enzymes in the pathway or in their activity. The most commonly seen is galactose 1 phosphate uridyl transferase deficiency, although galactokinase and UDP galactose epimerase deficiencies are also seen. And so it's important to catch this early and to switch the milk out of the diet and replace it with a milk substitute. This is generally soy formula or soy milk. Now, there are other monosaccharides that can enter this pathway, and another important one is fructose. Fructose can enter by two separate means. It can enter in at the level of fructose 1-phosphate. And what we're going to do is we're going to first phosphorylate fructose to trap it in the cell and to activate it. We're then going to split this molecule into two trioses. But because we only have fructose 1-phosphate, we're going to end up with a triose that has a phosphate group and a triose that does not have a phosphate group. The enzyme that carries out this reaction is fructose 1-phosphate aldolase. And the products are glyceraldehyde and dihydroxyacetone phosphate. Now, we have two products that we really can't use yet. We don't need glyceraldehyde, we need glyceraldehyde 3-phosphate. And so we're going to use a triose kinase to phosphorylate glyceraldehyde to yield glyceraldehyde 3-phosphate. This can then enter glycolysis in stage 3. And dihydroxyacetone phosphate can simply be isomerized by triose phosphate isomerase, which is the same enzyme we saw in glycolysis. And finally, we end up with two molecules of glyceraldehyde 3-phosphate from this one molecule of fructose. Again, the enzymes are fructokinase that does the phosphorylation of fructose to yield fructose 1-phosphate. This is broken down by fructose 1-phosphate aldolase to yield glyceraldehyde and dihydroxyacetone phosphate. Triose kinase phosphorylates glyceraldehyde to yield glyceraldehyde 3-phosphate, and triose phosphate isomerase isomerizes dihydroxyacetone phosphate to yield the second molecule of glyceraldehyde 3-phosphate, and then this can enter into stage 3 of glycolysis. Fructose metabolism differs in the adipose tissue. In the adipose tissue, fructose is phosphorylated by hexokinase. Now, this is because hexokinase can phosphorylate many different hexoses: fructose, glucose. However, it has a higher affinity for glucose than it does for fructose. And in most other tissues, hexokinase will be busy phosphorylating glucose. But in the adipose tissue, the molar concentration of fructose is much higher than that of glucose. And so, hexokinase will phosphorylate fructose to yield fructose 6-phosphate. This metabolite can then enter glycolysis through the normal pathway. Now, another catabolic metabolite is glycerol. Glycerol is yielded from the breakdown of triacylglycerol. This breakdown product can enter the glycolytic pathway at stage 2. This is at the level of dihydroxyacetone phosphate, which will be isomerized to glyceraldehyde 3-phosphate. So glycerol comes from triacylglycerols. These are lipids that consist of a glycerol molecule with 3 fatty acid chains attached. During the breakdown of fats, these fatty acids can be removed to yield glycerol. Glycerol enters into the glycolytic pathway in this manner. We start off with glycerol and then we phosphorylate it again to trap it in the cell and to activate it, so that when we end up with the end products, we'll have a phosphorylated activated metabolite. Glycerol phosphate is at the level of an alcohol, and we want a molecule at the level of an aldehyde. But we can't get there from here. We're going to start off with glycerol phosphate, and then we're going to oxidize it to form dihydroxyacetone phosphate. So we're going to form the ketone first, and then this will be isomerized to form the aldehyde that we want, glyceraldehyde 3-phosphate. So again, the enzymes that catalyze these reactions are glycerol kinase that catalyzes the phosphorylation of glycerol to glycerol phosphate, Glycerol phosphate dehydrogenase oxidizes glycerol phosphate to yield dihydroxyacetone phosphate. During glycolysis, the yield from one molecule of glucose is 2 ATP, 2 NADH, and 2 pyruvate molecules. In the process of glycolysis, 2 molecules of NAD+ are used for each glucose molecule. Because NAD+ is formed from niacin, which is an essential vitamin, generally the supplies in the cell of both NAD+ and niacin are limiting. And so if NAD+ supplies in the cell are not replenished, they will run out eventually. One of the ways to regain NAD+ is through the formation of ethanol in fermentation. This replenishes NAD+ stores so that glycolysis can continue. There are other methods of replenishing the stores of NAD+ in the cell. In fact, there are 3 major ways. 2 of these are anaerobic and occur under conditions of low or no oxygen. The third method is aerobic, and this occurs when oxygen is not limiting. Let's first look at the anaerobic pathways. One of them we saw briefly is the formation of ethanol. Now, the regeneration of NAD+ occurs in 2 steps during alcoholic fermentation. First, we have a decarboxylation of pyruvate to acetaldehyde plus carbon dioxide. The enzyme that does this is pyruvate decarboxylase. Again, the name suggests what the job is. Then we have the reduction of acetaldehyde to ethanol, coupled to the oxidation of NADH to NAD+. And so, NADH will be the electron donor to donate electrons to acetaldehyde to yield ethanol. Pyruvate can also be used to yield lactate. In this pathway, NADH reduces pyruvate to form lactate, and in the process, NAD+ is generated. Lactate is made in red blood cells, and it's also made in muscles that are working above the body's ability to provide oxygen to that tissue. The production of lactate occurs as a direct reduction of pyruvate. So we're going to take the 3-carbon molecule pyruvate and we're going to reduce that to form lactate. The enzyme that does this is lactate dehydrogenase. The electron donor again is NADH, and we're going to yield NAD+ plus lactate. And finally, in the aerobic pathway, we have a decarboxylation again of pyruvate, going from a 3-carbon pyruvate to a 2-carbon acetyl-CoA. Next comes further oxidation, and this occurs in the electron transport chain, in which electrons are ultimately donated to oxygen. Acetyl-CoA goes through the TCA cycle, and then the reducing equivalents that are formed during the TCA cycle go on to the electron transport chain, where they are oxidized. In this way, NAD+ is replenished by the electron transport chain, in which electrons are ultimately donated to oxygen. Now, an important thing to know is the Cori cycle. The lactate that is produced in the muscles and in red blood cells will enter the bloodstream and travel back to the liver, where it's used to make glucose in a process which we'll talk about during the next lecture, gluconeogenesis. Gluconeogenesis is an energy-consuming process, but by making glucose from lactate, you can supply the body's energy needs by providing glucose for the other tissues. And so again, when oxygen levels are low, glucose will be broken down to pyruvate, but then that pyruvate cannot go through an oxidative phosphorylation because there's insufficient oxygen. And so it will instead be reduced to form lactate. This lactate, rather than build up in the muscle, will be transported in the blood to the liver. And in the liver, lactate can be oxidized to pyruvate, and then pyruvate can be used to form glucose again. Some of the most potent pathogens that we know are obligate anaerobes. These include the bacteria responsible for tetanus, botulism, gangrene, cat scratch fever, and abdominal, pelvic, pulmonary, and blood infections. These must exist in an anaerobic environment. Now, E. coli that lives in our gut can function either anaerobically or aerobically depending on the conditions. This is known as being a facultative anaerobe. This bacterium is an anaerobic bacterium under low oxygen conditions, or it can grow quite nicely in an oxygen-filled environment as an aerobe. And so again, E. coli is a facultative anaerobe.